Good morning, children. Yesterday we did the glycosidic bond, which is present in carbohydrates. Now, and also we did the types of carbohydrates, monosaccharides, disaccharides, and polysaccharides. If you have any problem in that, I think you must have gone through it yesterday. If you have any problem in that, just let me know just now so that I'll repeat that topic for you. Or shall I proceed with the nucleic acid? Yes, beta. Any problem in yesterday's work? Yes, Pache. Okay. So I'll proceed with another one. So we have to study now the nucleic acid, the sugars, etc. Nucleic acid. Nucleic acid. We have two types of this. We have DNA and we have RNA. The full form of DNA and RNA, you all know. What is the full form of DNA? What is the full form of DNA? Nucleic acid. And what is the full form of RNA? Ribonucleic acid. Now that means where does the difference lies? Where does the difference lie? The difference is in the sugar. The deoxyribose sugar is present in DNA and ribose sugar is present in RNA. So we have deoxyribose sugar in DNA and we have ribose sugar in RNA. Another difference which is there is that if it is DNA, it will have T, nitrogenous bases. Have you heard of nitrogenous bases? So in that nitrogenous base, this DNA has thymine as the nitrogenous base, whereas in place of thymine, what is present in RNA is uracil, URACIL, which is represented by the letter U, and this is represented by the letter T. This is present in RNA. So here the difference lies between DNA and RNA. Both are nucleic acids, both are genetic material. And there is it is said that RNA was the one which came into existence first. It was RNA which came into existence first, and after that DNA came into existence because RNA is unstable. So RNA is considered to be unstable. So from that unstable RNA, DNA came, DNA originated, which was more stable, which was stable than RNA. So it is said that it is RNA which came into existence first, that is before DNA. And then DNA came later on. So there was a scientist. First, let me tell you two terms you will come across in your book, that is nucleoside and nucleotide. These are the two terms which you will come across in your book, nucleoside and nucleotide. Now, as I told you, both the genetic material, DNA and RNA, I'm not telling you anything just now about these two. I'm just telling you a general thing. We all know that DNA and RNA, both these genetic material, these are made up of sugar. And which sugar? If we say sugar, which sugar? Pentose sugar. Pentose. Pentose sugar. What is pentose sugar? Anybody who can tell me? What is pentose sugar? Yes, beta. What is pentose sugar? Who's going to tell me? Yes, pentose sugar. What is pentose sugar? Yeah. Yes, beta. Who will tell me what is pentose sugar? No? No? Okay. Pentose means 5. A sugar in which 5 carbons are present. 5. Pentose means 5. A sugar in which 5 carbons are present. So both in DNA and RNA, sugar is present. That means deoxyribose sugar and ribose sugar. And both are pentose sugar. That means in both 5 carbons are present. There is slight difference also in their structure. The ribose sugar, which is present in DNA, which is a pentose sugar, of course, is different from the sugar which is present in RNA. And this is ribose sugar, and it is also a pentose sugar. Both sugars, deoxyribose sugar, which is present in DNA, ribose sugar, which is present in RNA, both are pentose sugar. That means in both 5 carbons are present, but structurally they are little different. Now, besides this, we have the phosphate or inorganic phosphate. Phosphate is also present. Okay, phosphate. This is also present. And besides this, one more thing which is present, those are nitrogenous bases. Nitrogenous bases. These 3 things are present. Nitrogenous bases are 4 in number, and one is different in both DNA and RNA, which I have already written on the board, thymine and uracil. Now, these 3 things are present in DNA and RNA. That is why this sugar, which is a pentose sugar, phosphate and nitrogenous bases, this is present in DNA and RNA. Now, then what is meant by nucleoside and what is meant by nucleotide? If there is only sugar and nitrogenous bases, pentose sugar, I'm writing pentose sugar. If there is only pentose sugar plus nitrogenous bases, then it is a nucleoside. If there is pentose sugar plus nitrogenous bases, it is a nucleoside. We call it a nucleoside. But if there is pentose sugar plus nitrogenous bases plus phosphate, then we call it a nucleotide. If all the 3 things are present, that means if pentose sugar is present, nitrogenous bases are present, and phosphate is present, we call it a nucleotide. Now, this is that is why sometimes, you must have seen in DNA it is written DNA is a polynucleotide chain. For DNA it is written of course for RNA also and DNA also, polynucleotide chain. This is written DNA is a polynucleotide chain. Poly means many, nucleotides means the structures, these are present. So it is also known as polynucleotide. That means many of the structures are present. Now, how these structures are connected, this I'm going to tell you. First of all, I'll tell you what is a pentose sugar in both the cases. So the pentose sugar has a structure like this, 5 carbon sugar. So it is going to be like this. Okay. Here we have oxygen, carbon, 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 4th carbon, and 1 carbon is present here, which is in the form of this functional group, CH2OH. So this is a pentose sugar. Now to this pentose sugar, if it is a nucleoside, this is a pentose sugar, 5 carbon sugar, 1, 2, 3, 4, 5. Pentose sugar, only nitrogenous base is present. So this nitrogenous base is attached to 1st carbon. This is 1st carbon, this is 2nd, this is 3rd, this is 4th, and this is 5th carbon. So we have 5 carbons. I have underlined these 5 carbons. The nitrogenous base is attached here. So this becomes a nucleoside. Yes, this becomes a nucleoside. Now, how does a nucleotide appear then? Sorry, beta, it got displaced. I had to admit 2 students, Sarthak and Mannat. Yes, so this is a nucleoside, and nucleoside appears like this. Now, how does a nucleotide appear? So, nucleotide is like this. This is the pentose sugar, 5 carbon sugar, 1 carbon here. Okay, second carbon here, third carbon here, fourth carbon here, and the fifth carbon is in the form of CH2OH. And here we have oxygen. So this is pentose sugar. Again, to the first carbon, nitrogenous base will be added. Nitrogenous base is present, and there is one phosphate also. This phosphate is attached to the fifth carbon. This is the fifth carbon. So this phosphate is attached here. So this is the type of structure of a nucleotide. This is how a nucleoside looks, which has just pentose sugar and nitrogenous base. This is pentose sugar, which has 5 carbons plus nitrogenous base. And nucleotide appears to be like this. It is also it has also a pentose sugar. So this is 1, 2, 3, 4, 5 pentose sugar plus nitrogenous base attached to the first carbon and phosphate, which is attached to the fifth carbon. So this is the difference between nucleoside and nucleotide. Is it clear to you, children, or any question which you want, any query you have? Is it clear the structure of nucleoside and nucleotide, or any query you have? Okay, Amrita. Yes, others, beta. Any question, any query here? Okay, Ashika. Others, many are there. Siksha, fine, Siksha. Yes, yes. Okay, Tadar. Yes, others, only 3, 4. I don't want. I want more responses. Yes. Others, you could not understand. Always there are 3, 4 students who are responding. Yes, Khushman, Jasleen, Rohan, Lalit, Harsha, Sanvi, Ankita. Better you should also respond so that I come to know that you are understanding. That is why I asked, so that if you have a query or anywhere if you could not understand, okay, Sapak, I will repeat, no problem. I am here to teach you. I am here to teach you. If you do not understand, this is my duty to tell you. You can just come out with that doubt. Just say, ma'am, I could not understand here. Okay, Rohan, so you can go through the video afterwards. Fine. If you, or the net, because of network issue, you have joined late, no problem. Fine. So, fine. You can go through the video. I'll upload it. Okay, fine. Fine. So, Lalit, is it clear to you? Lalit, are you listening to me? Yes, Lalit. Lalit, I want your response. Okay. No response at all. So fine. So these are pentose sugars I have made with nitrogenous bases in nucleoside, pentose sugars with nitrogenous bases and phosphate in case of nucleotide. Now, the nucleosides and nucleotides, they have different names. How are they named? If you know, in nucleoside we have pentose sugar and nitrogenous bases. So I'll represent pentose sugar by PS. If we have PS, pentose sugar plus nitrogenous base. First, before this, I'll tell you what are nitrogenous bases. That also you should know that what are nitrogenous bases. So I'll tell you what are nitrogenous bases. Nitrogenous bases are 4 in number. These are 4 in number, and they are adenine. They are of 2 types first. Let me tell you, they are of 2 types. They can be purines and they can be pyrimidines. Either they are purines or they are pyrimidines. Now, there is difference between the purines and the pyrimidines. The difference between the two is, one is these purines, they are 2 in number. Their purines are 2 in number. I think you will not be able to see here. I'll write it at the top so that you are able to see. I'll write here again. Nitrogenous bases. These are of two types. Either they are purines or they are pyrimidines. Either they are purines or they are pyrimidines. Now, as I told you, purines are 2 in number. Example of the purines, they are only 2 in number, A and G. A here represents A and G. A means adenine, ADENINE, and G means guanine, GUANINE. These are 2, adenine and guanine. Pyrimidines are 3 number. Pyrimidines are 3 in number, and those 3 are C, T, and U. T is present in the DNA, U is present in RNA. This means cytosine, thymine, and U is uracil. So these are these 3 numbers. So nitrogenous bases are of basically these are two types, purines and pyrimidines. Purines are A and G, pyrimidines are CTU. So if we see the DNA, I'm writing here for a minute. If we see the DNA will have the 4 nitrogenous bases, A, G, C, and T, adenine, guanine, cytosine, and thymine. The RNA will also have 4 nitrogenous bases, and those 4 nitrogenous bases will be A, G, C, and in place of T, it will have U. So these 4 nitrogenous bases will be present in RNA, and these 4 nitrogenous bases will be present in DNA. So now another difference between the purines and pyrimidines. The purines are dicyclic. Bidicyclic means they have 2 rings, and pyrimidines are monocyclic. Monocyclic means, mono means single. They are single ring compounds. So one are dicyclic, another are monocyclic. Another difference between the two is the purines have 4 nitrogen. 4 nitrogen atoms are present in them. 4 nitrogen atoms are present. I'm just writing it together because the space is left. And here 2 nitrogen atoms are present. So there 4 nitrogen atoms are present here. 2 nitrogen atoms are present. Another difference is what is the position of that nitrogen atom. These 4 nitrogen atoms are present at first, third, seventh, and ninth position. The 4 nitrogen atoms are present at first, third, seventh, and ninth position. It has 2 rings. And they will be present in first, third, seventh, and ninth. Whereas these 2 nitrogen atoms are present in first and third position. These are present at first and third position. So these are nitrogenous bases, which are 4 in number, which are of 2 types, purines and pyrimidines. And this is the difference between the two. Any problem in this? Any doubt in this? Which you want me to repeat, beta? Which you want me to repeat? Yes? No? Clear, beta? This point is clear? Okay. Now, I was telling you something about what happens, the type of nucleosides and nucleotides. Because there was nitrogenous bases. So I just wanted to tell you before that what are nitrogenous bases. Pentose sugars, already I have told you that pentose sugars are the sugars which have 5 carbons in them, and these are the 5 carbons. Okay, now what is a nucleoside and nucleotide? How many types of they are? So I told you that I'll write only PS. If this pentose sugar is attached to, this pentose sugar is attached to the first nitrogenous base, let's take it adenine. If it is attached to A, then what do we get? We get adenosine. Adenosine. I'm not writing the full form of A because I have already written here adenine. A is adenine. So if pentose sugar is attached with adenine, we get adenosine. If pentose sugar is attached with guanine, we get guanosine. If pentose sugar is attached with C, we get cytosine. If pentose sugar is attached with T, we get thymidine. So these are, an uridine if it is attached with U. Pentose sugar with U, uridine. Okay, so these are these are the nucleosides which we get. These are the nucleosides which are present. I just want to show you a picture of monocyclic and dicyclic. You will have to wait for 1 minute because I'll show you that in the book, and I have to open the book for that. Just a minute, beta. I'm just want to show you something. Yes, here, if you see in this book, if you see this picture, this has 2 rings, one ring, another ring. So it is a purine. And this is just one ring. It is a pyrimidine. It is written here also, pyrimidine and purine. Adenine is a purine. So it has 2 rings. Okay, and it is a pyrimidine. It has just one ring. The position of nitrogen is first, third, seventh, and ninth position. Here it is just first and third position. Okay, so this is what I wanted to show you, that it appears to be like this. And now I would tell you, this is uridine. This is uridine. Now I would like to tell you what is a nucleotide. How does the nucleotide appear? Yes. So how do the nucleotide appear? This is what I want to tell you now. Now in this, if this pentose sugar is attached to the phosphorus is already there, it has to be there in nucleotide, plus let's take it adenine. If pentose sugar plus phosphate plus adenine, which nucleotide we get? We get adenylic acid. Adenylic acid. So this is the first uh, nucleotide, adenylic acid. Same way you can write yourself also, for pentose sugar plus phosphate plus G, we get guanylic acid. Same way pentose sugar plus phosphate plus C, citridylic acid. Pentose sugar plus P plus T, thymidyllic acid. And pentose sugar plus P plus U, uridylic acid. So these are the types of nucleotides, and that side, on the left side, I have written the types of nucleoside. I think this is clear to you. Better, this is clear to you? No doubt, no query? Is there any doubt, any query, any repetition if you want? Yes? Or shall I proceed to the next one? Clear. Fine, fine. So what I'll proceed to the next one is the pentose sugars. This is the last topic for today because the time is running out. So nitrogenous bases and nucleosides, nucleotides, you have already understood. So I'll show you the pentose sugars, the two types of pentose sugars. So the chemical structure of pentose sugars. Chemical structures of both the pentose sugars, the one which is present in DNA and the one which is present in RNA. The structure is like this. Yeah. Now, pentose sugar obviously means 5-carbon sugar, so it will be like this. Oxygen. Here we have